qualitative, so we should know first what is qualitative. So you're checking the quality. That means either the presence are there in the sample or not. Particular element or particular molecule or particular thing, whatever it is. If it is present, that means the quality is there. So the presence or the existence of the molecule. So in general, in qualitative, we'll get urine samples. So as per the curriculum, we got urine samples. The urine sample having two types, normal urine and abnormal urine. So in the normal urine, you will be having urea, uric acid and creatinine. So these are the components. So in that also you will be having physical you will be having something like how much volume, odor, and something called specific gravity. Specific gravity is nothing but the constituents, how many like solutes present in that will give you the specific gravity. So other than that, you will be like how it is appearance. Is it very transparent, clear? In few conditions like alkaptonuria and phenylketonuria, MSUD, your urine color will be something different. So in few cases like blood in urine and all these things. So when you see the appearance, the turbidity, if you're having proteins, there will be a turbidity. If you're having phosphate ions, there will be a turbidity. So appearance also matters here. So all these things comes under the physical examination. So when it comes to the chemical examination, so when you're doing the urine under the chemical examination, you can able to find out is it a normal urine or it's an abnormal urine. If it is a normal urine, you will be having urea, uric acid and creatinine. These are the components which are regularly present in that. So when it comes to the abnormal urine, so it's a thumb rule. There will be 6 components which are comes in a 3 sets. 6 components in 3 sets. What are the 6 components in 3 sets? First one, you will be having sugar or glucose plus ketone bodies. Sugar and ketone bodies, we will call it as glucose and ketone bodies. Second one, blood and protein. And the third one, when it comes to the third one, so it will be your bile salts and bile pigments. You can see that there are 6 components: glucose, ketone bodies, blood, protein, bile salts, bile pigments, but 3 sets. So there is a simple technique. When you got a sample, you don't need to worry that which one I got. It's a very easy thing. If you got something transparent, remember that that's a glucose and ketone body. If you got something reddish in color, I will show you in the video also. A reddish in color, you can say that blood in urine, blood and protein present in that. So if you got light yellowish or green, you can say that that's bile salts or bile pigments are present in that. So these are the basic things. So in qualitative, you're going to do these things. So you have to identify these things. Even you're doing this glucose and ketone bodies, blood and protein, bile salts and bile pigments, each and every set. So this sample having separate tests, this one is having separate tests, this one is having separate tests. You need to do 6 tests. In that, in examination, you will get any one sample. That means your 2 tests should be very perfect. Remaining 4 you can manage. You can do something and you can show that, sir, these are absent. But remember that if you got a red color sample, that means the blood and protein is present. If you're giving your benzodine is absent, that's blunder mistake. Don't do those things. If you got something like bile salts and bile pigments, if you're going with your pouches with a normal color, that's a blunder mistake. Don't do that. Remember that you got the color, first to do this thing. If I got, so if I'm doing the practical, I got a slight yellow greenish color sample. I need to do first. So I used to do bile salts and bile pigments first. Then rest of the 4 I'll do perform and I'll keep those things aside. When they ask, sir, I did all the 6, but I got these 2 positive. You can save the time. You don't need to rush. You don't need to remember anything else. When you got, see, I'll tell you the some tip in this. If you got Viva also, they'll ask you, if I got bile salts and bile pigment, they'll ask those things only. Which one is called as bile salt? Which one is called as bile pigment? In between the practical, you can get the suggestions from your friends. If you know answers, that's good. If you don't know also, you can get good with these things. You can know. So you can ask them what is bile salt, what is bile pigment, which metabolism will give you bile salt, which metabolism will give you bile pigment. So in which conditions you can see the excretion of these things in the urine and all. So in my clear video, I'll tell you that, but it's better if you don't know also, just at the time of practical, you ask them. So whatever the sample you got, if you got sugar and ketone bodies, you should know. You should know what is the normal range of sugar, what is the abnormal range, in which condition you can see sugar in the urine, in which conditions you can see the ketone bodies in the urine. For example, diabetic ketoacidosis. In these conditions, you can see sugars as well as ketone bodies very high in the blood. So sometimes when they got more than the renal threshold, they will be excreted through the urine. So in these conditions, you can see very easily. So there are thumb disorders, thumb rules for these things. You need to follow that. If you can answer in that manner what they're asking, if you're giving them a correct answer, you're going to secure 16 to 18 marks. If you're giving damn good, you're going to get 20 out of 20. So we are going to discuss about how this abnormal urine will be discussed. Majority colleges will give this abnormal urine only. Very few colleges will do this urea, uric acid, creatinine. This is very easy to do, but this is somewhat tricky. You need to do some homework about these things. So the first one, glucose. If you want to identify what is a glucose, it's a simple thing. You need to do a test called UBT, urine Benedict's test. The Benedict's test you're doing normally is a sample for serum and all they used to do. So or else some normal sample they used to do. Here what you're doing is urine Benedict's test. What you're going to do, you're taking the urine sample. You're going to take the urine sample and you're going to add the Benedict's reagent. Upon heating, you're going to get color change. So initially it is blue in color. Which one? Benedict's reagent. The Benedict's blue in color will combine with the urine sample. Upon heating, you're going to get color. It can be from orange to brick red based on the percentage of sugar. This is you might have studied already, but I'm giving you this as outline. So based on the color, the concentration will be there. If you're having more sugars, it will be a brickish red color. So this is what the thing happens. If you're doing the ketone bodies, you can go with the Ruthra's test. Upon Ruthra's test, when you're doing this, sample will be in, it's a ring between the junction of two layers. For Benzidine, you will be having a bluish color. So for Ruthra's, when you do the Ruthra's, so when you're doing this, you can be able to see that upon adding the acid, you can see that there is a ring between the junction of two layers. That indicates the presence of ketone bodies. So when you're doing Benzidine for the blood, so it will turn into greenish blue color. So when you do protein, what you're going to do, you're taking the test tube. So you're going to fill the half of the test tube and you're going to heat the upper corner. When you heat the upper corner, protein will get denatured. Formation of coagulum, that is called proteins. So when you're doing the bile salts and bile pigment, bile salts is very easy to identify. You can do that sulfur test. So we call it a Hayes sulfur test. What happens? The surface tension will determine here. So you're going to add, you're going to use a control, you're going to use a test. In the control, you're going to take the distilled water. In the test, you're going to take the sample. In the distilled water, you're going to add the one spatula of your sulfur powder. It won't sink because of that surface tension and all. When you add that same sulfur powder in the solution, that means urine sample with the bile salts, allows your sample to sink. So that indicates the presence of bile salts in the urine. When it comes to the bile pigments, when you do the filtration and all, the filtrate will be there in the paper, filter paper. On the filter paper, when you add the Faucher's reagent, you can see the color change, greenish color change. If by that you can able to identify that the bile pigments are present on that. With this, the qualitative you can manage the best. So in detailed video of this will be continued. For more content, tricks and tips of biochemistry and other subjects of your first year MBBS, just do follow us, subscribe, and you can follow us in the Instagram, ABC-Biochemistry, and MedMemes, who supports and content regular uploads. You can go through with these two. Thank you.